Hey everyone, welcome to this explainer. Today we're diving straight into something absolutely mind-blowing, CRISPR. This isn't just some small incremental step forward in biology. We're literally talking about scientists rewriting the very blueprint of life, fundamentally shifting how we interact with the natural world. Let's get right into it.
Okay, let's look at this massive number right here, 3.2 billion. Just imagine a giant instruction manual that contains every single tiny detail on how to build and operate, well, you. That manual, it's exactly 3.2 billion letters long. That is just an astronomical amount of biological data crammed into every single cell of your body.
Now think about this. What if, buried in those billions of letters, there's a tiny typo? Just one wrong letter out of 3.2 billion. But that single little error causes a massive, life-altering genetic disorder. For basically all of human history, we were totally stuck with whatever manual we were born with, typos and all. But ask yourself, how does the whole picture change if we suddenly get our hands on molecular scissors? What if we could scan that entire manual, pinpoint that exact typo, and just replace it with the right letter in seconds? That right there is the fundamental promise of CRISPR.
Section 1. The Blueprint of Life. Understanding the code. So, as you guys probably know, DNA is our foundational code. We've got those four chemical letters, A, T, C, and G, arranged into specific sequences we call genes. Think of them like precise sentences in our manual that give instructions for literally everything, from your eye color to how your body pumps out insulin. But here's the catch. Every single time your cells divide, this massive 3.2 billion letter code has to be copied. And, well, biology's copying machine isn't exactly flawless every time. Which brings us to Section 2, the mutation problem and accumulating errors. Now, what's really wild to see here is the sheer scale of the clutter. Sure, our cells have ways to fix some errors, but a lot of copying mistakes, or mutations, just kind of stick around and build up over millions of years. Because of this, roughly 85% of our DNA is just totally non-functional junk data. It's kind of like old, obsolete files completely clogging up your computer's hard drive. The vast majority of our genome is just packed with broken instructions, pseudogenes, and old viral DNA segments we've just dragged along through our evolutionary history.
Before CRISPR, trying to find and fix one specific functional gene hiding in all that junk data, it was a monumental nightmare. Someone once described it as being like trying to intercept an intermediate-range ballistic missile traveling at 20,000 kilometers per hour. Before these recent breakthroughs, it was incredibly difficult, super clunky, and honestly, completely lacked precision. Nailing the exact genetic target was basically impossible.
All right, Section 3, the molecular interceptor, precision editing.
Okay, so think of CRISPR like a highly advanced biological missile defense system for your genome. In the pre-CRISPR days, genetic engineering was essentially like flying first-generation manual jet fighters, clunky, analog, zero-targeting radar. But the CRISPR era, it's like rolling out a sixth-generation fighter fully hooked up with AI. It can effortlessly navigate the insane maze of our DNA, totally ignore that 85% of junk data, and lock straight onto the exact gene that needs a patch. It's a total game changer.
Let's actually break down how this molecular interceptor does its thing in 3 simple steps without getting bogged down in the chemistry. Step 1, just like modern defense systems use radar to spot a threat, CRISPR uses a guide molecule to scan and find a specific sequence of DNA. It's literally a genome radar. Step 2, once it locks onto the target, it deploys a protein called Cas9 to act as the interceptor. And then step 3, that Cas9 protein makes a super clean, ultra-precise molecular cut right at that exact location. Find, intercept, cut. Actually, it's just an incredibly elegant piece of biology. Let's move to Section 4, rewriting biological history, the impact. The real-world applications here are honestly mind-bending. We're not just talking about tweaking some superficial traits, we're talking about taking genetic diseases and basically editing them right out of our biological history. Going back to our aviation analogy, it takes us from just putting band-aids on a glitchy aircraft to automatically correcting the plane's biological stability, much like a modern fly-by-wire system. But it goes way deeper than that. Could we reprogram humans to have pre-birth immunity to certain diseases? The potential is absolutely there. We can even recode agriculture, so crops become climate resilient. You know how natural microevolutions led to things like antibiotic-resistant bacteria? Well, with CRISPR, we're the ones steering the wheel now.
Which inevitably leads us to Section 5, the ethical dilemma in exponential consequences.
So as objective observers here, we really have to impartially weigh two massive realities. On one hand, you have scientific realism, the very real, life-saving ability to fix terrible genetic disorders and build resilient agriculture to feed a changing planet. But on the flip side, we crash straight into some massive ethical dilemmas. Are we talking about engineering designer babies? What happens when we make permanent, irreversible changes to humanity's collective blueprint? The line between simply curing a disease and actively enhancing a human being is incredibly blurry, and it's a tightrope society is going to have to carefully walk.
And the thing is, this isn't happening in slow motion. CRISPR technology is advancing at an absolutely exponential rate. It's just like that old fable, putting one grain of rice on the first square of a chessboard and doubling it every square after. It very quickly explodes into this impossible amount. Our ability to handle the consequences, the ethics, and the safety of this whole shebang absolutely has to keep pace with the technology itself.
I'll leave you with this final provocative thought to chew on today. Staying informed is step one to grasping this massive revolution happening right now, literally inside our own cells. But as we make the leap from just being passive readers of our genetic code to being the active editors, are we truly ready for the profound responsibility of rewriting the blueprint of life? Thank you so much for joining me for this explainer, and keep exploring.